So, good morning to all of you. Yesterday, we have tried to understand about the cell. Specifically, uh, basically, what we have discussed yesterday uh, was the introduction to cell. What is cell? Cell theory, shape, size, and other aspects of the cell. And then we have also discussed how to go about the study of the cell, where we have decided to study the cell under four headings. For general cell, and if it is only the animal cell, under only three headings. There is the first one being the cell wall, then cell membrane, then comes the cytoplasm, and fourth one being the nucleus. This is what we are expected to study. So today we will start our discussion with this cell membrane, or what is known as the plasma membrane. Very, very important, even from the examination point of view also, especially the structure and function of this plasma membrane is very, very essential. When we talk about this cell membrane, plasma membrane, or in short it is also known as plasma lemma, this is the outermost boundary of the animal cell. But whereas if it is a plant cell, this is next to the cell wall. This cell membrane or plasma membrane is a very, very unique organelle of a cell. Not because it is the outermost boundary of the animal cell, but because of the intricacies as well as the function that it carries. It is one of the very, very important one. Basically, this plasma membrane or cell membrane is invisible with our light microscope. None of us will be able to observe this in the laboratory using the light microscope, or what you call the compound microscope, because it is so thin, that is 7 to 8 nanometers in thickness, which all of you know will be able to see that only with the help of electron microscope. If you see that using the electron microscope, it is appearing like a railway track. Probably you have seen the railway track. Two metallic bars run parallel to each other. In between there will be railings which connect them. So it appears like that. The very, very speciality of this plasma membrane is selectively permeable. It is a selectively permeable membrane. So when I say what do you mean by this selective permeability? Anything can be of two types, or sometimes three types. Either it may be impermeable, any membrane which doesn't allow the movement of any solute molecule across it, or it is completely permeable, it can allow anything and everything to pass through it, what we call permeable, or it can be semi-permeable, which can allow the movement of certain molecules of some specific size. In that way, plasma membrane, many scientists say that, earlier they used to say that it is a semi-permeable membrane. But in my opinion, it is not semi-permeable, but it is selectively permeable. That is, it can sense, it has a brain of its own, by means of which it selects what are the materials that are to be allowed through it or not. It is very, very selective as far as the permeability is concerned. So that's why it is known as selectively permeable membrane, because few molecules move very freely through the plasma membrane. For example, water, carbon dioxide, ammonia, and oxygen, almost all gases, without any hindrance, without any obstruction, they can move across this plasma membrane from inside to outside and outside to inside. But there are many things which cannot pass through the membrane easily, still they are pass through it because of the some proteins which are embedded in the plasma membrane, what we call the carrier protein. These proteins which are embedded in the bilayer, I will come to that, and they help in the transport of certain material across the membrane. What material? That has been selected by the plasma membrane. In order to understand this, how the plasma membrane becomes selectively permeable. This is a very, very interesting thing. If a membrane is permeable or non-permeable, it has a definite physical property which do not change. But whereas here in the plasma membrane, it is not so. The physical properties of this plasma membrane also changes so as to facilitate the movement of certain material when and where and when it is required. Now the question is how the plasma membrane is able to get this property. What makes the plasma membrane selectively permeable? Actually, for this, we have to understand the structure of the plasma membrane. Basically, understanding the structure of the plasma membrane was very, very difficult. For this, first, in order to understand the structure, we should know about the composition. What are the basic composition of a plasma membrane? If you look into the plasma membrane, the basic composition consists of lipid, phospholipids, some proteins, cholesterol, glycoproteins, lipoproteins. These are the composition of a plasma membrane. Not composition, chemical composition of a plasma membrane. Now the question is how these chemicals are arranged, configured so as to make it selectively permeable. This was a mystery. Even though you know this, the composition, chemical nature, now the very mysterious thing is to know what makes it selectively permeable. In order to achieve this selective permeability, these glycolipids, phospholipids, glycolipids, cholesterol materials, other integral proteins are to be arranged in certain or should be fixed in a fixed pattern so as to make it selectively permeable. So many scientists try to find answer for this, and there are different models have also been proposed, like unit model, unit membrane model and all, but none of them were practical enough to explain the selective permeability of this plasma membrane. It was only the one model, what is known as fluid mosaic model, proposed by Nicholson and Singer, helped us in understanding the architecture of this plasma membrane. The name is given fluid mosaic model. We will see why it is called so. As per this model, you can see, or only this model helps us in exchange or movement of the material, which is selective movement, I should say. See, uh, some of the small molecules which can pass through easily, and other molecules are, some are lipid soluble and water soluble, and they should also move. All these things were explained by this fluid mosaic model. Okay, before understanding the arrangement or architecture of the plasma membrane, we should also know about the membrane proteins. I have told you there are integral proteins as far as the chemistry of this plasma membrane is concerned. One is phospholipids, second one is the glycolipids and glycolipids, and then cholesterol material. There are number of proteins. Most of the proteins are the integral proteins. See, this is the plasma membrane where you can see number of such proteins. Even cholesterol materials are also there, that I will come to. In these proteins, basically there are two types. Channel proteins. Channel proteins, see, these are the channels. And another one is the receptor proteins. Receptor proteins are mostly the peripheral proteins, whereas the channel proteins are the integral proteins. What is the function of the receptor proteins? The surface of the receptor protein, which project slightly out of this plasma membrane, when it comes in contact with the chemicals or other molecules, it can recognize them. That is the role of this one. Then glycoproteins, the protein which is associated with glucose. Glycoproteins, they identify the different cell types. They are also peripheral, mostly attached to this integral protein. You can see the glycoproteins. Along with that, there are few proteins which function as an enzyme. Probably all of you know the famous saying that all proteins are, all enzymes are proteins. So that's why here there are many proteins which function as an enzyme, and that catalyzes the production of substances. So these are four important components along with the cholesterol molecule that is found in the plasma membrane. See, the plasma membrane, that is being the boundary of the cell, it has two walls. One is the outside wall and this is the inside wall. Outside wall, which is facing outside, and it is having having the power to communicate with the neighboring cell. The cell wall of this cell communicates with the cell wall of the next cell. That is the outside. Whereas the inside wall of the plasma membrane always communicates with the cytoplasm, because the plasma membrane forms the boundary to the cell and it surrounds this cytoplasm. Now here, this plasma membrane, the major component is phospholipid bilayer. There are two layers. See, this is one layer of phospholipid and this is another layer of phospholipid. It is made up of two layers of plasmolipid. Thus, it forms the boundary. Then another important function of the plasma membrane, because in order to understand the structure, we should know what are the roles that are played by the plasma membrane is equally important. One is it forms the boundary. it protects the boundary, it encloses or encircles the cell. That's one important thing. Then the second important function of the plasma membrane is to regulate what enters and what leaves the cell. So that's why I have told you it is selectively permeable. It has to select what are the materials which can go out, what are the materials which can come inside the cell. That means it has a regulatory role of what enters and what leaves the cell. In addition, it also provides support and protection. Now, if you look into the structure of the plasma membrane, what is there is mostly it is made up of lipid bilayer, that is the phospholipid bilayer, and proteins are embedded within that. So this model or this structure of the plasma membrane, which satisfactorily explain the selective permeability, was given by Nicholson and Singer in 1972. And this model is known as fluid mosaic model. Why it's fluid? The plasma membrane itself is fluid in nature. Its fluidity is there. Then it looks mosaic. What do you mean by mosaic? I hope all of you must have seen the mosaic flooring. On the floor, you fit the tiles of different colors. On the floor, you fit the tiles of different colors, and that give you a mosaic appearance. ಗೊತ್ತಿದೆಯಾ ಈ ನೆಲಕ್ಕೆ ಕೆಲವೊಂದು ಸಾರಿ ಟೈಲ್ಸ್ ಹಾಕಬೇಕಾದ್ರೆ ಬೇರೆ ಬೇರೆ ಕಲರ್ ಟೈಲ್ಸನ್ನೆಲ್ಲ ಫಿಕ್ಸ್ ಮಾಡಿ ಅದನ್ನು ಫಿಕ್ಸ್ ಮಾಡಿರ್ತಾರೆ, ಅದಕ್ಕೆ ಮೊಸಾಯಿಕ್ ಟೈಲಿಂಗ್ ಅಂತಾರೆ. ಸೊ ಹಿಯರ್ ಆಲ್ಸೋ ದ ಅರೇಂಜ್ಮೆಂಟ್ ಈಸ್ ಮೊಸಾಯಿಕ್ ಬಿಕಾಸ್ phospholipid cells, in between integral proteins, peripheral proteins, on the surface, glycolipids, isn't it? So that's why if you observe that from the top, it looks like a mosaic floor. And it is fluidic. So that's why the model that has been proposed by Nicholson and Singer in 1972 is known as fluid mosaic model. And this is the only model which satisfactorily explains the selective permeability and all other functions of this plasma membrane. Now, as far as this fluid mosaic model, there's a double layer of phospholipids. There are two layers of phospholipids. And a phospholipid molecule, if you see, a single phospholipid molecule, how it appears is, it is having a single head and two tails are hanging from the head. And the properties of the head and tail are different. Head is hydrophilic, that is the water loving, whereas the tail is hydrophobic, water hating. Hydrophilic heads are facing outside. Hydrophilic tails are inside. That means out of the two layers, the head is facing outside, and in between that two heads, the tails of both the phospholipids are buried. In between these phospholipid molecules, integral proteins of different types are embedded. It's also true if I say proteins and other molecules are embedded in a framework of phospholipid. Actually, basic structure is formed by the phospholipid. And within this framework of phospholipid, proteins and other molecules are embedded, giving a mosaic appearance. mosaic appearance. So that's why it is known as mosaic model. A membrane is fluid, I have already told you. This is because for the fact that most proteins and phospholipid molecules, they move laterally. They are not fixed, they move laterally. As a result of the movement of these proteins and phospholipid molecules, the membrane is fluid. The phospholipids are the most abundant lipids in the plasma membrane. Major part of the plasma membrane is formed from these phospholipids. These phospholipid molecules are empipathic molecules because the same molecule at head region it has one type of property, at the tail region it has a different type of property. Empipathic. The head is hydrophobic. And it is containing both hydrophobic as well as hydrophilic, water hating as well as hydrophobic means water hating, hydrophilic means water loving. Both the reasons are there. Again to say, the membrane is a fluid structure because of the lateral movement of these phospholipid molecules and the proteins. It's also mosaic because various proteins are embedded and that gives the appearance of a mosaic. See here, this is the phospholipid molecule, this is the head, this is the tail. And in the plasma membrane, they are arranged like this. There are two layers of phospholipid. This is one layer and this is another layer. And these phospholipid molecules are arranged in such that the head is facing outside and tails are buried inside. And now, in between that you can see different types of integral proteins. These are the integral proteins. So that's why when you look it from the top, it looks like a mosaic pattern. And all these things can move. This can go move and come here. This can move and go somewhere here. This can come here because of the lateral movements. So that's why it is showing the fluidity. Then on the surface, if you look, in addition to these phospholipid molecules and the integral proteins, there are many glycoproteins, glycoproteins, glycolipids, cholesterol molecules here. See, all those things are possible. These phospholipids in the plasma membrane can move within the bilayer. Within the bilayer. Most of the lipids and some proteins, they can drift laterally, oscillate from here, they can push and go somewhere. But very rarely, the molecules can flip-flop transversely across the membrane. See, this can move to here, this can move to somewhere here. That's the lateral movement, that is drift lateral. Sometimes they can flip-flop. This molecule can jump here and this can jump here. This can jump to this layer and the one which is here can jump to this. That's a flip-flop. This is the lateral movement from here to here. Approximately 10 raised to 7 times per second. But flip-flop is very, very rare. At maybe once per month like that. In a very cool temperature. The membrane switch off the fluid state to a solid state. The reason is very simple. If the temperature drops, the membrane solidifies. At what temperature the membrane solidifies depends on the type of the lipid, because all of you know that lipids or fats, they become solid in the low temperature, which all of you might have seen that, isn't it? That's the property of the lipid. So suppose if you have the coconut oil, how it appears in the winter. Coconut oil is a fluid, but in the winter early morning if you look into it, that gets solidified. The same thing happens because major component is the phospholipid. But this fluidity also varies depending upon the fatty type of the fatty acid. If the membrane is rich in unsaturated fatty acids, they are more fluid than the membrane which are rich in unsaturated fatty acids. But for the functioning of the membrane must be fluidic. If the membrane solidifies, it will not be able to achieve this selective permeability. See, in case of the unsaturated hydrocarbon tails, which kinks and it becomes more fluidic. See, kinks and it becomes more fluidic. Whereas saturated hydrocarbon tails, it becomes viscous. In low temperature, it becomes more fluidic, and it becomes less fluidic, and high temperature it becomes more fluidic. And the same membrane becomes viscous during the low temperature. I have already told you there are some cholesterols. Cholesterols are steroids. They have different effect on membrane fluidity at different temperature. The cholesterol material is also embedded within the phospholipid. At warm temperature, such as 37 degrees Celsius, cholesterol restrain the movement of phospholipids. It packs. Otherwise, what happens? The membrane may become more volatile, may show more movements. But at cool temperature, the same cholesterol maintains the fluidity by preventing tight packing of the tails. Still, it allows the tails to kink. Otherwise, at cool temperature, the membrane will become solidified, and when the cell membrane becomes solidified, it will not be able to carry out any work. So, but it is avoided even in cool temperature by means of having the cholesterol material in between, which prevents the tight packing of the phospholipid molecules, thus maintaining the fluidity. See the cholesterol molecules, and this is the phospholipid, the head, and this is the tail. This is also head. Tails are buried inside, heads are facing outside. In between that, this cholesterol material is concerned with the physics of these phospholipid molecules. Some of these proteins can also drift in the plasma membrane within the bilayer. They can float within the plasma. And proteins are much larger than the lipids, because all of you know proteins are the macromolecules. They move very slowly. Whether they move slowly or fast, it is not the thing. It is What counts is the fluidity. So the change, it changes the architecture. The plasma membrane has lot of functions to carry out. Basically, as I have told you, the plasma membrane is a collage of different proteins. It's a collage of different proteins embedded in the fluid matrix of the lipid bilayer. What do you mean by collage? Collage is an art. ಗೊತ್ತಿದೆಯಾ? ಪೇಪರ್ ಪೀಸು ರದ್ದಿ ಇದನ್ನೆಲ್ಲ ಇದು ಮಾಡಿ ಕಟ್ ಮಾಡಿ ಅದನ್ನೊಂದು ಶೇಪ್ ಕೊಟ್ಟು ಅದಕ್ಕೆ ಒಂದು ಥೀಮನ್ನು ಡೆವಲಪ್ ಮಾಡ್ತಾರೆ. ದಟ್ ಈಸ್ collage. So a collage, it's a collage where different proteins, phospholipids, cholesterol, glycoproteins, all are involved in the fluid matrix of bilayer, and it gives you a perfect picture. The proteins, the type of the proteins within the plasma membrane determines the function of this plasma membrane. Whereas the peripheral proteins, they have the function of recognizing the signals or other molecules and all those things. The integral proteins, which penetrate in the tail region, the hydrophobic core, and they are often span the entire membrane. See here how they are arranged. It's a collage. It's a perfect collage. See, from the top or even from the integral proteins, peripheral proteins, carbohydrate, glycoproteins, which already we have seen. The integral proteins that span the membrane are called transmembrane protein. They are also called as transmembrane protein. In the transmembrane proteins, there is an hydrophobic region of one or more stretches and contain the amino acids, where the most of the amino acids are coiled, and there is the coiling is of alpha helical. See, the transmembrane proteins, they show the amino acid as it shows the alpha helix. There are 6 major functions that are assigned to plasma membrane. One is transport, second one is enzymatic activity, signal transduction, cell-to-cell recognition, intercellular joining, and attachment to cytoskeleton and extracellular matrix. This is the list of the functions. I will explain each and every one. So the first one, transport, I will keep it later. First, many of the plasma membranes function as an enzyme. What are enzymes? Enzymes are the biocatalysts which are used to enhance the biochemical reaction without undergoing any change in the reaction. So now, see, this is the enzyme, this is the proteins. It can convert, this is a substrate, it fixes with the enzyme and get converted into an intermediate product. Again, that is taken by another enzyme. It finally converts into a useful end product. This is by means of biochemical reaction. And being the students of biology, you know, the process or this entire process of biochemical reaction can be accelerated in the presence of enzymes because they are biocatalysts. All of you know, being the student of chemistry also, how the catalyst works. These are the biocatalysts. see, that can convert the raw material into product. Then another important function of the plasma membrane is cell to cell recognition. It should be able to recognize the neighboring cells as self, its own. Many times the cell fail to recognize the neighboring cell as the cells of the same body. In that case, it may lead to autoimmune disorders. So, how the cell recognize each other by binding to the surface molecules on the plasma membrane. Then the membrane carbohydrates may bind covalently with the lipids. But what's important is the carbohydrates which are found on the external side of the plasma membrane vary among the species, individuals, and even the cell type in an individual. So that's why it becomes easy for one cell to recognize the other one. If there's a variation, it will not accept it. It will recognize it as some foreign body, foreign material. Very important in developing our immune system. So, even these membranes are very distinct. The membrane of each cell is distinct, both inside as well as outside faces. How? It all depends on the distribution of proteins, lipids, and their association with the carbohydrate in the plasma membrane. So this is very important. That is providing uniqueness to each of these cell. The plasma membranes are synthesized from endoplasmic reticulum and Golgi apparatus. During that time only, there will be an asymmetrical distribution of this protein, lipid, and associated carbohydrate, which make each plasma membrane unique and helps in identifying. This is how the plasma membrane is produced. So this membrane structure results in the selective permeability. It controls or regulates the exchange of materials, and thus it regulates the cell's molecular traffic. Water, gases can pass easily. But whereas some of the polar molecules, non-polar molecules can pass easily, like hydrocarbons, because they can dissolve in the lipid bilayer and pass through the membrane. But whereas the polar molecules such as sugar and other things do not cross the membrane very easily. In the beginning of the class, if you recollect, I have told you, there are some transport proteins which allow the passage of the hydrophilic substances across the membrane. There are other type of protein, what you call channel proteins, which have some hydrophilic channels, and through this tunnel or channel, certain molecules can pass. Such channel proteins, which are embedded in the plasma membrane, they are called as aquaporins, which helps in the passage of water. There are other proteins also, which are called as carrier proteins. Here the role of enzyme comes. They bind to the molecule, they change their shape and shuttle across the membrane. Otherwise, in their original shape, they cannot be. They are large enough not to allow them inside or to allow them to go outside. But these carrier proteins, when they bind with the molecules, they bring some conformational changes to their shape and makes them to shuttle between the or shuttle across the membrane. A transport protein is very specific for the substance it moves. One type of transport protein or carrier protein will not be able to help for the other molecule. Only of that type of molecules, specific molecules, can be transported across the membrane. How this transport is achieved? One is no energy investment. Simple diffusion, which all of you know. Diffusion is the tendency for molecules to spread out evenly into available space from the region of higher concentration to the region of lower concentration. They spread out evenly into the available space. Although each molecule may move randomly and the diffusion of a population of molecules may exhibit, but the net movement will be in one direction, that is from the region of higher concentration to the region of lower concentration, till the dynamic equilibrium is attained. Till the dynamic equilibrium is attained, this will continue. So this is the process where energy is not required. See, here the concentration is more. So from the region of higher concentration, this has moved to the other side. Net diffusion. How long? When the concentration of this region and this region becomes same, this movement continues. This is what is called as diffusion process. There is no requirement of energy. That means the substances diffuse down their concentration gradient. No energy is utilized here. So that's why it is known as passive transport. And there is no need of energy investment. This is what? Just to rebrush about the functions of plasma membrane, I have told you: selective permeability, healing, phagocytosis, pinocytosis, cell-to-cell recognition, perception of stimuli, reception of signals, influx or efflux of ions and nutrients. See, this is the diffusion process, how it happens. From the region of higher concentration to the region of lower concentration, you can call it as the diffusion. Without energy expenditure. But every time you cannot expect the free transport. Sometimes the molecules are to be transported from the region of higher concentration to the region, from the region of lower concentration to the region of higher concentration, which involves the investment of energy. For this, the protein pumps are there. Energy is required. Here you can see these are integral protein. For one molecule, for example, here you have the sodium. Sodium is have to be moved inside. How? See, using the active transport system. Energy intensive, wherein which there are protein pumps, protein channels, which function as protein pumps. They will transport them. But diffusion is very simple. See, from the region of higher concentration to the region of lower concentration. But it's a passive transport. But active transport, there is a need for energy. Then exocytosis and endocytosis. exocytosis. Suppose there are materials which are to be sent outside, some of the proteins which are of no use inside the cell. Then also they are to be moved out. For this, the plasma membrane has found out a very new phenomenon. What it does is, it encircles this material which are to be sent outside with a membrane, and this membrane is similar to that of the plasma membrane, and this is what you call vesicles. This vesicle in the cytoplasm is moved toward the plasma membrane. Once this touches the plasma membrane, at the region of the touch, the plasma membrane dissolves like this, and it tries to amalgamate this vesicle, fuse this vesicle. See, once the vesicle is fused, the membrane which is surrounding the vesicle also breaks and pushing the material outside the cell. See, this inside, it has been bound in the vesicle. Vesicle moves to the plasma membrane, fuses with the plasma membrane, and then pushes out the material. So when there is a bulk transport, mostly it does with the vesicle, because Golgi body form the vesicle, and this Golgi body vesicles merge with the plasma membrane and release their content. Even this is how the neurotransmitters are released at the synaptic junction. Endocytosis is also the same phenomenon. See, the material which are to be moved in by endocyte, they come to the plasma membrane, and plasma membrane itself forms a vesicle form and pushes that vesicle inside. Once it comes inside, the vesicle disintegrates and the materials are free to move in the cytoplasm. So here the plasma membrane sinks inwards, pinches off and forms the vesicle. Sink, form a vesicle and pinches off. Vesicle or again merges with Golgi, and Golgi body starts storing the carbon. Even plasma membranes show phagocytosis, cell eating, if it captures the solid particle. Phagocytosis is shown by the amoeba, where pseudopodia encloses the things and takes inside. So, sometimes bacteria, cell debris or food, by that it can eat the solid food. Even the same type liquid food can be taken. It is what you call pinocytosis or cell drinking. Endocytosis already I have told you. Well, see how the receptors, and it forms vesicles and release the content inside. This is how the mechanism. See, materials, vesicles are taken care of. So here, before winding, I would like to show you the one small video. Probably it will be of much useful to you in understanding the. It's of 1 or 2 seconds. Sorry, 2 minutes video, I think. Slideshow. See, which you can see how the fluid mosaic model of the plasma membrane, which you can see here. So there are so many cells in our body, and all of you know the DNA is the special cell machinery. This is the plasma membrane. See, the what you call the fluidity to the plasma membrane. The inside cell, there is the outside cell, and many materials can pass through the cell. The cell membrane, this is the structure of the cell membrane, 3D structure, where you can see is the top view. So if you look from the top, it looks like a mosaic, and in between that there are integral proteins from the side view. Fluid mosaic model. See, they can move phospholipids. This single phospholipid molecule. See, hydrophilic head and hydrophobic tail. This is how it's a lipid bilayer, how these phospholipid molecules are arranged. Water lovers and water haters. See, both inside and outside the cell there is a plenty of water. In between that there are cholesterol molecules, which is more important for the viscosity of this membrane. And this is the integral proteins, peripheral proteins, and glycoproteins. Integral proteins are mostly the carrier proteins which have a channel inside and facilitate the movement of molecules, which are selectively permeable. That means that selectively permeable. See, inside and outside, and this is the how the materials move against the concentration gradient. Some of the material can pass through directly through the plasma, this phospholipid bilayer. So, this is what I wanted to discuss as far as the plasma membranes are concerned. And now tell me whether you have understood the things or not. And ಹೇಳಿ. Yes. Yes. ಪೂರ್ವಿಕಾ, ಏನು ಅರ್ಥ ಆಯ್ತಾ ಇಲ್ವಾ? ಹಲೋ, ಸರಿ ಸರಿ ಕೇಳ್ತಾ ಇತ್ತಾ? ಏನೂ ಪ್ರಾಬ್ಲಮ್ ಇಲ್ಲ. ಯಾವ ದೆರ್ ವಾಸ್ ನೋ ಪ್ರಾಬ್ಲಮ್ ಇನ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ದ ಥಿಂಗ್ಸ್. ಅದೇ ಸೊ ವಿಡಿಯೋ ಇದೆಲ್ಲ ಸರಿ ಕಾಣಿಸ್ತಾ ಇತ್ತಲ್ಲ? ಓಕೆ. So then we will meet in the next class. Now we have completed the plasma membrane, then we will enter into the cytoplasm. Okay? Fine, thank you.